Bismillahirrahmanirrahim. Welcome back, friends. Welcome to my channel, Kami Biology. My name is Muhammad Kamal Khan, and today in this video I'm going to talk about the difference between sense, antisense, coding, non-coding, and template strand. Most of the students don't know about the sense strand, antisense, coding, non-coding, and template strand. They don't know about that, and they make a lot of mistake in by identifying each of the strand. So today this video is especially for those students who don't know about the strand, okay, that which one is the coding, non-coding, and anti-template strand. So let me start. So what are these strands and how we can identify each of the strand in a DNA sequence, in a DNA? So let's explain. So now let me write that we are going to talk about okay. Now how we will identify that? For understanding the coding, non-coding, antisense, and sense strand, let me write some of the sequences to you guys. So let's suppose these are some of the sequences: G, A, T, C, A, G, C, T, A, and they have also a direction to 3 prime, and here will be the 5 prime. Okay. Now remember, when the examiner asked from you that, let's suppose you have the strand and they have also a direction 5 to 3 prime, and when he asks from you that make a complementary of that strand, then it is very easy for us. How you will make the complementary? You can make the complementary of that strand by just make the opposite of this strand. When you make the opposite of this strand, it will be the complementary. For example, this strand have 5 to 3 prime direction, so the complementary strand have 3 to 5 prime direction. Okay. Now what are the complementary of this one strand? The complementary is that we are making to opposite of that strand, means guanine. So we better know about that, if we want to make a complementary of the strand, so we better know about that adenine will always make a bond with thymine. Cytosine will always make a triple bond with guanine. So as you can see, this upper strand is the strand. So how you will make the complementary of that strand? We should know about that guanine will make a bond with cytosine, adenine will make a bond with thymine, thymine will make a bond with adenine, cytosine will make a bond guanine, then thymine, cytosine, guanine, adenine, and thymine, and the direction will be also opposite, means 3 prime to 5 prime. This is how you can make the complementary. Okay. So if the teacher asks from you that how you will make the complementary strand, so it is very simple for us. Just we should know about that guanine make a bond with which of the nucleotide. We should know that adenine make a bond with which one of the nucleotide. We should know that thymine will make a bond with which one of the nucleotide, and similarly cytosine will make a bond which one of the nucleotide. We should know about that. Then if the teacher asks from you that make the RNA sequences of this one strand, if your teacher asks from you that make the RNA sequences according to the upper strand, then how you will make that? You will simply make that guanine, here you will also write guanine, adenine. Now adenine will replace with uracil. Sorry, you will also write here adenine, thymine will replace with uracil, cytosine, guanine, you will also write cytosine, adenine, guanine, cytosine, uracil, adenine. And all of you better know that RNA will have always 5 prime to 3 prime end. This is very important. RNA have always 5 prime to 3 prime end. As you can see, upper strand have always 5 to 3 prime end. Now if you want to make the RNA sequences according to that strand, then you will not make any kind of the changes in the sequences in the RNA. You will just make a change at the thymine. You will just replace the thymine with uracil, as you can see. So here I will just replace the thymine with uracil. and the remaining sequences are the same. Okay, if the direction is 5 to 3 prime, then you will write the same sequences which is present at this one strand, but just you will replace the thymine with uracil. But if your teacher asks from you that make a RNA sequences of this one strand, then how you will make that? Again, in this case, it is very simple. So similarly, we can also make the complementary sequence, means we will also make the RNA sequences according to that. So here will be C, T, sorry, T will be replaced with U, A, G, U, C, G, A, U, and they have 5 prime to 3 prime direction. Is it right? Many of the students said that yes, it is right, but it is wrong. It will be wrong. How? You are, means I already told you that we have the same sequences, but just we are, I also replace the uracil with thymine. So how I say that it is wrong? Every means, the sequences are the same, but in RNA sequence we have uracil. So I already read the result. Then how I say that it is wrong? Still it is wrong. Why? Because in this case, just concentrate on the direction. Direction is 3 to 5 prime. And I already told you that the RNA direction will be 5 to 3 prime. 5 to 3 prime the direction. So if you write the direction 3 to 5, it will be wrong. And if the direction is not right, so definitely the sequences will be also not right. You writing the sequence means the sequences is right, but the position of each sequences are not right. So if the teacher asks you that make the make this RNA sequences according to that the lower strand, then you will make the complementary of the lower strand. It is very important. Okay, you will make the complementary. Now once you make the complementary, and let's suppose here you are making the complementary of this one strand. For example, C will make a bond with guanine, T will make a bond with adenine, A will make a bond with uracil. In this case we have uracil, and then guanine will make a bond with cytosine, adenine, guanine, cytosine, uracil and adenine. So as you can see, we make a complementary of this one strand. So it is a complementary. And now just compare. So this one strand is also similar and this one strand is also similar. Now remember, when the teacher asks from you that make the RNA sequences of this strand having the direction is 3 to 5 prime, then you will make the complementary. One thing, you will make the complementary. The second is that you will replace also the thymine with uracil. Okay. But if the teacher asks to you that make a complementary, if he asks from you that make the RNA sequences of this one strand having 5 to 3 prime, then you will write the same sequences, but just you will, replace the thymine with uracil. This is very important. You will always, make the strand like that as I explained to you. Okay. Now, which one is the coding and which one is the non-coding? Which one is the sense and which one is the antisense? Remember, if the strand have 5 to 3 prime direction, this strand will be called as coding strand. And we also call that sense strand as well. But if the strand have 3 to 5 prime end, it will be called as non-coding strand. Or it also called antisense. Okay. Similarly, RNA strand will always the coding strand. RNA strand, as you can see, it have the 5 to 3 prime end. That's why it will always the coding strand. And we also call it sense strand because it will give a sense to the protein synthesis. And the upper strand is also called the coding strand having 5 to 3 prime end because the RNA are matching with the upper strand. Just thymine is not matching, but the remaining sequences are matching. That's why we can say that the upper strand of the DNA having 5 to 3 prime end and the RNA strand are always called as coding and sense strand. But the lower strand of the DNA is called as template strand and it is called as non-coding strand and antisense. And we have the RNA polymerase enzyme mainly RNA polymerase 2. It will responsible to make the RNA sequences by using the lower strand as a template. Why it use the lower strand and why it not use the upper strand? The reason behind that, it use the lower strand because the lower strand have 3 to 5 prime end and RNA will always synthesize in 5 to 3 prime end. That's why it will use the lower strand. And moreover, lower strand have anti-codon, means they have all the anti-coding sequences, this lower end. So that's why RNA will always make a coding sequences. Remember, if the upper strand have already coding sequences, if the upper strand have already sense, if the upper strand have already 5 to 3 prime direction, then why the RNA using this one strand? RNA will not use this because this strand have already 5 to 3 prime direction. This strand have already all the coding and everything is fine, which we need in the RNA sequences. That's why it will, most of it will use 3 to 5 prime because it need to make the coding. So once it need to make the coding, so it need the non-coding strand. Okay, so that's all about the coding, non-coding, anti and non-anti. So you need to understand about two things here. The two thing is that you need to know about the direction. If the direction is given to you, then you can easily make the complementary. If the direction is given to you, you can easily make the RNA sequences. So here, one thing we should know about the direction. Once we get idea about the direction of the strand, then we can easily make the complementary, we can easily make the RNA sequences. We can easily identify the coding, non-coding, antisense strand. But if the direction not given, most of the time the direction, they will always give you the direction. If the direction is not given to you, then this strand is, let's suppose the direction is not given to you, then this one strand is also called coding and non-coding as well, because due to this strand you can make the complementary, and due to this strand you can also make the what we can say RNA sequences. So when you make the RNA sequences, it will be non-coding. When you make the complementary, it will be also the coding, because you can make a complementary, it will be also coding. Okay, so most of the time they will always give you the direction. So you need to fully focus on the direction. So I hope you guys get idea about that, what is the coding, non-coding, and how you will identify each strand in a DNA molecule. So I hope you get idea about that. But if still you have any kind of question in your mind, you can write it into a comment. Inshallah, I will give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video and subscribe the channel to get more interesting videos like that. Thank you so much for watching.